Hello friends, today we are going to start the solution of GATE 2021 Engineering Science paper in which we are discussing food technology sections. Topic is microbial growth and date kinetics. Please subscribe the channel Neer Education. Question is, in a typical bacterial growth curve, the first order kinetics for growth rate is observed in option is lag phase, log phase, stationary phase, death phase. Correct answer is log phase. That means in a typical bacterial growth curve, in log phase, first order kinetics for growth rate is observed. Now we will see the discussion. There are four types of phases in bacterial growth curve cycle. As you can see in this figure, this is the figure of bacterial growth curve that represents the number of living cells in a population over time. This plot is plotted between the log of number of bacteria and incubation time, in which the four phases lag phase, exponential or log phase, stationary phase and death phase are indicated with plot. In lag phase you can see there is a horizontal plot, that means the bacterial growth is negligible, whereas in exponential phase bacterial growth occurs in exponential manner, exponential phase. In stationary phase, the rate of growth of bacteria is equal to the rate of bacterial population, so that it seems the bacterial population constant. Whereas in dead phase, the bacterial population decline at exponential rate. Next is in lag phase, when microorganisms are inoculated into fresh culture medium, usually no immediate increase in cell number occurs. As we can see in the previous plot, there is no increase in the cell number of bacterial population, because that's why the plot is in horizontal direction in lag phase. Therefore, this period is known as lag phase. This lag phase is also known as tooling up phase, because in this phase the cells prepare themselves for growth in the new medium. Second one is exponential or log phase. In this phase, the growth observed is balanced growth, wherein the cells are dividing regularly by binary fission and are growing by geometric progression. In this phase, the rate of growth of bacterial population is constant, and this rate of growth is expressed as generation time, also known as the doubling time of the bacterial population. Next one is the stationary phase. In this phase, the bacterial population growth stops and the growth curve becomes horizontal, as you can see in previous plot. This occurs as the number of cells dividing would equal cell death and the number of viable microorganisms remains constant. That means in this phase the rate of growth of bacterial population is equal to the declining rate of bacterial population. That means it seems that viable microorganisms population remains constant. That's why there is a horizontal plot in this stationary phase. Fourth one is declining or death phase. In this phase, the if incubation continues after the population reaches a stationary phase, a death phase follows, in which the viable cells population declines. In this phase, viable cells population declines, and during this phase, the viable population declines geometrically or exponentially, essentially the reverse of growth during the log phase. We can see in the plot that in log phase the bacterial population increases exponentially, whereas in death phase the bacterial population decline exponentially. That means the in death phase it is the reverse of the uh, plot of log phase. Okay, thank you.